The receptor, tyrosine, and kinase, and cancer. So the receptor tyrosine kinases are highly affinity cell surface receptors for many polypeptide growth factors, cytokinins, and hormones. And of the 90 unique tyrosine kinase genes identified in the human genome, 58 encode receptor tyrosine kinase proteins. So the receptor tyrosine kinases, which is really hard for me to say, have been shown not only to be key regulators of normal cellular processes, but they also to have a clinical role in development and the progression of many types of cancer. So the mutations in the receptor tyrosine kinases lead to activation of a series of signaling cascades which have numerous effects on protein expressions. So they are part of a larger family of proteins. They encompass the receptor proteins, which contain a transmembrane domain, as well as a non-receptor, which they do not, the non-receptor does not possess transmembrane domains. So a tyrosine kinase is an enzyme that can transfer a phosphate group from an ATP to a protein in a cell. It functions as an on-off switch in many cellular functions. So they are a subclass of the protein kinase, and the phosphate group is attached to the amino acid tyrosine on the protein. So tyrosine kinases are a subgroup of the larger class of protein kinases that attach phosphate groups to other amino acids like serine and 309. So phosphorylation of protein by kinases is an important mechanism in communicating signals with a cell. And this communicating is what we call signal transduction and regulating cell activity such as cell division. So the protein kinases can become mutated, stuck on the on position, and cause unregulated cell growth, which is necessary for the development of cancer. So there are some kinase inhibitors such as imatinib, and they are effective cancer treatments. So most tyrosine kinases have an associated protein, tyrosine phosphate, which removes the phosphate group. And protein kinases are of a group of enzymes that possess a catalytic subunit that transfer the gamma or the terminal phosphate from the nucleotide triospates, and that's usually ATP, to one or more amino acid residues in a protein substrate side chain that results in change affecting the protein's function. So the enzymes fall into two broad categories, characterized by respect to their substrate, specifically the serine 309 specific and the tyrosine specific. So the kinase is a large family of enzymes that are responsible for catalyzing the transfer of phosphoryl group from a nucleotide triphosphate donor such as ATP to an acceptor molecule. So the tyrosine kinases catalyze the phosphorylation of tyrosine residues in proteins. And the phosphorylation of tyrosine residues in turn causes a change in the function of the protein that can be that they are contained in. So phosphorylation at tyrosine residues controls a wide range of properties in proteins such as enzyme activity, subcellular localization, and interaction between molecules. So tyrosine kinases function in many signal transduction cascades where extracellular signals are transmitted through the cell membranes to the cytoplasm, often the nucleus, where the gene expression may be modified. And finally, mutations can cause some tyrosine kinases to become constitutively active, a nonstop function state that may contribute to initiation or a progression of the cancer. So the kinases function in a variety of processes. It's the pathways, the actions, and they're responsible for the key events in the body. The receptor tyrosine kinases function in transmembrane signaling, where the tyrosine kinases with the cell function and signal transduction within the cell function in signal transduction to the nucleus. So the tyrosine kinase activity in the nucleus involves the cell cycle control and the properties of transcription factors. So in this way, the activity is involved in mitogenesis, or the induction of mitosis into a cell. Proteins in the cytosol and proteins in the nucleus are phosphorylated at tyrosine residues during this process. So cellular growth and reproduction may rely to some degree on the tyrosine kinases. And the function has been observed in the nuclear matrix, which compromises not the chromatin, but rather the nuclear envelope, and a fibrous web that serves to physically stabilize. And if we wanted to get really specific, the LYN, a type of kinase in the SRC family, was identified in a nuclear matrix. It appears to control the cell cycle, and the SRC family tyrosine kinases are closely related but demonstrate a wide variety of functionality. So the roles of expression of the SRC family tyrosine kinases are very significantly according to cell type, as well as during cell growth and differentiation. So the LYN and the SRC family tyrosine kinases in general have been known to function in signal transduction pathways. So there is evidence that the LYN is located at the cell membrane, and it's associated both physically and functionally with a variety of receptor molecules. Fibroblasts, this is a type of cell that synthesizes the extracellular matrix and collagen, and it's involved in wound healing. That's been transformed by a polyomavirus process, higher tyrosine activity in the cellular matrix. So the tyrosine activity has been determined to be correlated with cellular transformation. And it's also demonstrated phosphorylation of a middle T antigen, or tyrosine, that's associated with cell transformation, a change that's similar to cellular growth and reproduction. And the transmission of a mechanical force or regulatory signals are quite fundamental in the normal survival of living organisms. So protein tyrosine kinase plays a role in this task too. And it's been called PP125. That's likely at the hand of the influence of the cellular focal of adhesions, and it's indicated by an immunofluorescent localization of that kinase. And local adhesions are not micro, but macromolecule structures that function in the transmission of mechanical force and regulatory signals. So among the scientific community, the PP125 is also referred to as FAK, which is focal adhesion kinase, because of its presence in the cellular focal adhesions. It's one of the major phosphotyrosine containing proteins in unaffected avian and rodent fibroblast cells. So fibroblasts are a cell that are responsible for wound healing and cell structure in animals, and among a number of other relatively minor but important jobs that take place occasionally. So this sequence, the PP125, when compared to national research, it's kind of unique, meaning that it could be a new member of the protein tyrosine kinase family. And the protein tyrosine kinase is up to about 70% unique compared to other protein tyrosine kinases. And that's a figure that's between actual members of an established protein tyrosine kinase family. So the amino acid sequence was observed indirectly, and it signifies association with the cytoplasm, dubbing it one of the large groups of cytoplasmic protein tyrosine kinases. So most changes are sometimes induced when the tyrosine kinase enzyme is affected by other factors. And one of the factors is a molecule that is bound relatively by a protein called a ligand. And you can see that in the diagram there. So a number of receptor tyrosine kinases, though certainly not all of them, do not perform protein kinase activity until they are occupied or activated by at least one of these ligands. And some research indicates that the receptors remain active within the endosomes, and it was once thought that endocytosis caused ligands was the event responsible for the process in which receptors are inactivated. And the activated receptor are internalized. That means they're recycled back into the system in a short time, and they're ultimately delivered to the lysosomes, where they become work adjacent to the catabolic acid hydrolysis that partake in digestion. So internalized signaling complexes are involved in different roles in different receptor tyrosine kinase systems, and the specifics of which were research that we just talked about. So ligands participate in reversible binding, which inhibits binding non-covalently, and this is the inhibition of different types or affected depending on whether these inhibitors bind the enzyme substrate, the complex, or both. Multivalency, which is an attitude that bears particular interest, is involved in scientific research, is a phenomenon characterized by concurrent binding of several ligands positioned on one unit to several coinciding receptors on another. So in any case, the binding of the ligand to its partner is apparently owing to the effects that it can have on the functionality of many of the proteins. And the ligand activated receptors are sometimes referred to demonstrate a unique attribute. So once it is bonded to the ligand, it is able to bind to the tyrosine kinase residing in the cytosol. And there are approximately 20 different receptor tyrosine kinase classes that have been identified. Some of the bigger ones are class 1 are the EGF receptors, or the ERB family. And I'm going to just start calling that RTKs. So most RTKs are single subunit receptors, but some exhibit a multimeric complex. The insulin receptor that forms disulfide linked dimers in the presence of hormones. So the ligand binding to the extracellular domain induces the formation of the receptor dimers. So each monomer has a single hydrophobic transmembrane spanning domain composed of 25 to 38 amino acids, and extracellular N-terminal region and an intracellular C region. So the extracellular terminal region exhibits a variety of elements, including immunoglobulin, like or epidermal growth factor like domains, and fibronectin type 3 repeats, or cysteine-rich regions that are characterized for each subfamily of RTKs. So these domains contain primarily ligand binding site, which binds extracellular ligands to a particular growth factor or a hormone. And the intracellular C-terminal region displays the highest level of conservation and compromises catalytic domains responsible for the kinase activity of these receptors, which catalyzes receptor autophosphorylation and tyrosine phosphorylation of RTK. The diagram that's shown here are the effects that I was just talking about of insulin. Okay, when you look at the slide, the top one is your phosphate, the middle is your ATP, and the bottom is your tyrosine. So in biochemistry, a kinase is a type of enzyme that transfers phosphate groups from high-energy donor molecules such as ATP to specific target molecules, which are substrates, and the process is termed phosphorylation. The opposite, an enzyme that removes a phosphate group from a target, is known as a phosphatase. So kinase enzymes that specifically phosphorylate tyrosine amino acids are termed tyrosine kinases. So when a growth factor binds to extracellular domain of an RTK, its dimerization is triggered with other adjacent RTKs. And the dimerization leads to a rapid activation of the protein cytomoplastic kinase domains, the first substrate for the domains being the receptor itself. And the activated receptor, as a result, then becomes autophosphorylated on multiple specific intercellular tyrosine residues. So through diverse means, extracellular ligand binding will specifically cause or stabilize receptor dimerization. And this allows a tyrosine in the cytomoplasmic portion of each receptor monomer to be phosphorylated by its partner receptor, propagating a signal through the plasma membrane. So the families. We have the epidermal growth factor receptor family. The ERB that we talked about earlier, protein family or epidermal growth factor family, is a family of 4 structurally related receptor tyrosine kinases. And if you have insufficient ERBB signaling in humans, it's associated with the development of neurodegenerative diseases such as multiple sclerosis or Alzheimer's disease. So they looked at mice, and there's a loss of signaling by any member of the ERBB family. It results in embryonic lethality, which defects to organs like the lungs, the skin, the heart, and the brain. And if there were excessive ERBB signaling, it's associated with the development of a wide variety of types of solid tumors. So the ERBB1 and 2 are found in many human cancers, and their excessive signaling may be critical factors in the development and malignancy of these tumors. Then next, we've got the fibroblast growth factor receptor family. It compromises the largest family of growth factor ligands at 23 members, and the natural alternative splicing of the 4 fibroblast growth factor receptor genes results in the production of over 48 different isomers. And these isoforms vary in their ligand binding properties and kinase domains, but they all share a common extracellular region composed of 3 immunoglobulin-like domains, and therefore they become like this big immunoglobulin superfamily. So interactions with them and their domains can be activated by several FGFs, and the FGFs themselves can also activate more than one receptor. And that's just a couple of the families. Okay, so the receptor tyrosine kinases pathway is carefully regulated by a variety of positive and negative feedback loops. And because the RTKs coordinate a variety of cellular functions such as cell proliferation and differentiation, they must be regulated to prevent severe abnormalities in cellular functioning such as cancer and fibrosis. So protein tyrosine phosphates are a group of enzymes that possess a catalytic domain with the phosphotyrosine specific phosphohydrolase activity. So these PTPs are capable of modifying the activity of a receptor tyrosine kinase in both a positive and a negative manner. The PTPs can dephosphorylate the activity phosphorylated tyrosine residues on the RTKs, which leads to termination of the signals. And studies involving these have shown that the regulation of the cell cycle and the cytokinin receptor signaling has dephosphorylated the epidermal growth factor receptor and the insulin receptor. So some PTPs, on the other hand, are cell surface receptors that play a positive role in cell signaling proliferation. Okay, our RTKs have become an attractive target for drug therapy due to their implication in a variety of cellular abnormalities such as cancer, degenerative diseases, and cardiovascular diseases. The United States Food and Drug Administration, FDA, has approved several anti-cancer drugs caused by the activated RTKs. So drugs have been developed to target the extracellular domain or the catalytic domain, and therefore they inhibit the ligand binding receptor oligomerization. Herceptin is a monoclonal antibody that is capable of binding to the extracellular domain of RTKs, and it's been used to treat HER2 overexpression in breast cancer. So if you look at the drug therapy on this chart, some molecule inhibitors and monoclonal antibodies, these are the ones that are approved by the U.S. Food and Drug Administration against RTKs for cancer therapy. You can go down through here and you can see the small molecules. You can see the targets. You can see which disease. You can see the renal cell carcinoma and the year that it was approved in 2005. Lung cancer, colorectal cancer. There was approval for this drug in 2004, and esophageal cancer, there were treatments that were approved in 2003 and 2004. So these are the cell signaling by the receptor tyrosine kinases that's having a huge impact on our medicine today, especially in treating things that have to deal with cancer. I probably used too many references because I needed to look up a lot of things on this. So I have 3 different slides. I'll try not to go too fast. But these are the works that I cited to find all of my information. I also gleaned a few things. Some of my references I actually just took one thing from, but I needed to look to understand it. And here is my 3rd slide. Thank you.